Hi everyone. Yeah, in the last class I was discussing about the infection. In that, how the microbial agents gets transmitted and what are the different sources for the microbial agents were discussed. And in this particular topic, I would like to discuss about how these microbial agents can evade the immune system of the host and responsible for their pathogenicity. And these are known as determinants of pathogenicity. And this determines how they escape from the host defense mechanisms uh, will be discussed now. And what are all the determinants and factors responsible for the evading this host defense mechanism will be discussed in this particular class. And the bacterial evasion from the host defenses will occur the first one by means of adhesion. Adhesion means attachment. And the bacteria will get attached to the surfaces, the cell surfaces, by means of fimbriae, which are external or cell wall appendages, also known as pili. These fimbriae are present in E. coli. Not only in E. coli, there's so many other organisms also will have the fimbriae around on the cell wall. And sometimes the adhesion also possible by non-pilous adhesions, which are known as glycocalyx, which is a polysaccharide slim layer produced by the Staph aureus. And the lipoteichic acid and M protein, that is a type of protein in the Streptococcus pyogenes, all are responsible for attachment of the bacterium to the cell or the infected cell in the host. And what is the importance of this fimbriae in adhesion and their important role in their pathogenicity? If you see, these fimbriae are antigenic in nature, and not only that, the adhesion will prevent the flushing away by the mucus secretions. When the bacteria or microbial agents gets attached to the mucosal surfaces, the flushing action of these mucosal secretions, this will be prevented by the bacterial adhesion. And once they get adhered, the microcolonies will be formed. Unless the bacteria produces microcolonies, they can't establish on their surface to produce the toxins or enzymes to responsible for their clinical manifestations. And this adhesion also facilitates the penetration of the microbial agent into the host cells. And the next one is invasiveness of the bacterium or microbial agent also determines their pathogenicity. What is the meaning for invasiveness? This is nothing but ability of an organism to invade the host cells after they get attached to the cell surface. And how is it possible? There are so many factors are there. The first one to be discussed among them is antiphagocytic factors. That means the cells or the agents which are responsible for antiphagocytic activity. That means they are microbial agents not amicable for the phagocytosis. Thereby they can invade the host cells. The first one among them is capsule, which is present in the pneumococci, M protein, which is present in the streptococcal cell wall, and cell wall protein A, which are present in the staphylococci. All these acts like antiphagocytic factors, thereby they are not destroyed by the phagocytosis of the phagocytic cells of the host, and they can multiply and invade the host cells. And some of the cytotoxins like hemolycins and leukocidins produced by the bacteria also responsible for invasion of the bacterium. Certain bacterial surface antigens like VA antigen seen on Salmonella species and K antigen present on Enterobacteria members of Enterobacteria family also acts like an antiphagocytic factors, so they can escape from the immune system of the host. And another thing is outer membrane protein, which is present in the gram negative bacteria, responsible for release of cytokines like interleukin 1 and tumor necrosis factor alpha, and they are responsible for their pathogenesis. And some of the enzymes like hyaluronidase enzyme, collagenase enzyme, kinases, coagulase enzyme, and IgA proteases. Like if you see the hyaluronidase and collagenases are produced by the streptococci, and coagulase produced by the Staphylococcus aureus. IgA proteases produced by the respiratory pathogens, and also even gonococci also produces IgA proteases. And these IgA antibody which protects the mucosal surfaces will be destroyed by the protease enzymes produced by the pathogenic bacteria, and they evade the mucosal surface immunological functions of the host. And the bacterial evasion from the host defenses. Another one is biofilm formation. What is the meaning for biofilm? So, well organized microcolonies of bacteria enclosed in a self-produced glycocalyx. So this glycocalyx, otherwise called extracellular polymer matrix, produced by the bacterium, and this bacteria will get protection from this glycocalyx from the environment. And Staphylococcus aureus and Staphylococcus epidermidis produces central venous catheters and contact lens infections or biofilm formation. And in case of dental plaque, the Streptococcus viridans is responsible for biofilm formation. And in case of respiratory tract, among the people who are suffering with the cystic fibrosis, the Pseudomonas aeruginosa produces biofilm, and this biofilm results in inherent resistance to drugs due to delayed penetrability of the antibiotic given for the purpose of treatment. And here you can see the biofilm formation. In the stage 1, you can see the planktonic bacteria adhere to the surface, which are present in the fluids going through the pipes or cannulas. And the sessile bacteria or individual bacteria begin to secrete the extracellular substance, that is glycocalyx. See, first they get attached, pink one. Once they gets attached, they produces the glycocalyx material. And in the third stage, you can see the development of this material and forms a film around the bacteria, which is known as biofilm. And this biofilm will protect the bacteria from the environmental defense mechanisms. And the fourth stage of this biofilm, and once it reaches the certain size, it gets detached from the particular area, and the microbial agents will get released into the blood circulation or to the environment, at that particular local environment, causes for the infections. And the intracellular survival, infected agent can overcome the action of phagocytic cell, but this by different means. In case of Mycobacterium tuberculosis and Legionella, they inhibit the phagolysosome fusion followed by the entry of the microbial agent or foreign cell into the phagocytic cell. And even though they entered the cell, some of the bacteria like Mycobacterium leprae are resistant to the lysosomal enzymes produced by the phagocytic lysosomes, and some of the bacteria will adapt to the cytoplasm of the phagocytic cell, and they get replicated. In case of Rickettsia and Listeria, which are intracellular organisms, they enter to the cell and they get by means of phagocytosis, and they get adapted to that cytoplasmic environment and gets replicated. And other intracellular organisms like all viruses, histoplasma, cryptococcus, which are fungi, plasmodium, leishmania, and trypanosomes, which are parasites, all are intracellular organisms which can evade the first defense mechanism of the host, that is phagocytosis, and gives the way for the microorganisms to multiply and to produce the disease. And some of the bacteria can produce the toxins, and these toxins responsible for their pathogenicity. There are two types of toxins will be produced with the bacteria. One type of toxins are known as exotoxins. Other type of toxins are known as endotoxins. Let us have the differences or properties of these exo as well as endotoxins. Exotoxins are proteins in nature and secreted by the bacteria, which are heat labile, and the production usually coded by the plasmids, which are extra chromosomal genetic elements present in the cell. And these are having enzymatic action, and exotoxins act specifically on certain cells, and the large doses are fatal. Not even small doses are not fatal, but large doses are fatal. And these exotoxins are highly antigenic in nature, and the resulting antibodies most of the times will give protection to the individual. And these exotoxins would be neutralized by antibody, and they could be toxoided. Toxide means when you expose the toxin to certain chemical agents, they loses their pathogenic property and retains their antigenic property, and those are known as toxoids. And these toxoids could be used as vaccines like tetanus toxoid, and the other usually the tetanus toxoid is the one which is commonly used in this particular purpose. And these exotoxins are produced by the gram positive bacteria, except the Shigella, cholera, and enterotoxigenic E. coli. Even though they are gram negative bacteria, they can produce exotoxins. Now we will see the endotoxins, which are lipopolysaccharides in nature, and they are not separately secreted by the bacteria, and they are nothing but part of cell wall, which are heat stable and produced by the bacterial chromosomes. And these endotoxins mediated by interleukin 1 and tumor necrosis factor, and these endotoxins acts each and every cell without any specificity. Even small doses are fatal in nature. They are less antigenic in nature and not neutralized by antibody, and they can't be toxoided and mostly produced by the gram negative bacteria. And exotoxins, some of the examples, there is a big list of toxins and a big list of microorganisms are there. But I am not going to confuse you people with the list of these toxins. And if you would like to learn, you can learn from the textbook. But here I would like to mention few exotoxins which are highly dangerous to the mankind. There is Corynebacterium diphtheria, which produces diphtheria toxin, and this toxin is responsible for respiratory infection and also the myocarditis in the infected individuals and cause for the death. Bacillus anthracis produces anthrax toxin, also is a fatal in nature. Clostridium tetani, which produces tetanus toxin, also responsible for a dreadful disease, that's tetanus. Clostridium botulinum, this produces botulinum toxin, which is responsible for severe foodborne disease and which is also fatal. Another thing is Vibrio cholera, which produces cholera toxin, causes for severe watery diarrhea. And I also mentioned about the endotoxins. Let us see what are the mechanisms of endotoxin producing the disease. One is they activates the macrophage and produce interleukin 1, interleukin 6, tumor necrosis factor alpha. All are responsible for the production of fever and inflammation. By means of the inflammatory process, they can cause for all the clinical manifestations. Next one is activation of alternative pathway of complement, responsible for the production of complement components like C3A and C5A, and which are responsible for the fever production and also promotes the inflammation, and again responsible for the shock by producing severe form of hypotension. And endothelial, platelet, and mast cell activation also will be seen by the endotoxic activity. And this mast cell, once they get activated, And these mast cells are activated by the IgE antibody, and this IgE antibody activates the mast cell, and they're finally responsible for increased vascular permeability, which is very important activity of the mast cell due to the degranulation of the mast cell followed by the IgE antibody. And activation of the coagulation pathway, which activates the Hagman factor and responsible for disseminated intravascular coagulation. And this disseminated intravascular coagulation causes the multi-organ failure, results in death of an individual also. And the special mechanisms of pathogenicity by the microorganisms. One is the pathogenic islands, and these are sets of genes present in bacterial chromosomes which are encoding for several virulence factors. These kind of pathogenic islands are seen in Shigella, Salmonella, Vibrio cholera, Yersinia pestis, Staph aureus, and uropathogenic E. coli. And the plasmids, which are extra chromosomal genetic elements, as long as these plasmids are present in the bacterial cell, they act like pathogenic agents. Once the plasmids are not seen in this bacteria, they never become pathogenic organisms. So these plasmids are responsible for the toxin production. For example, in E. coli, which produces enterotoxin, Staphylococci also produce enterotoxin. E. coli usually produces urinary tract infections, and Staphylococci produces wound infections. But because of the production of enterotoxin with the presence of plasmid responsible for that particular kind of toxin, this E. coli also responsible for their diarrheal diseases, and even Staphylococci also cause for diarrheal diseases. Another thing is superantigens, some of the toxins produced by the bacteria and some of viruses themselves will act like superantigens and causes for their pathogenicity. These superantigens are responsible for non-specific activation of T cells, which results in autoimmune-like response by producing large amounts of interleukin 1 and interleukin 2, which finally results in loss of specific T cell clones. When the specific T cell clones are not produced, all unwanted and abnormal antibodies will be produced, and they are responsible for the pathogenesis. And the terminologies commonly used in relation to the infectious agents are uh, virulence and the pathogenicity. So before going to talk about this virulence and pathogenicity, let us have the taxonomical views of these infectious agents. Usually the microbial agents, I am not going to the kingdom or family level. Usually we talk about the genus level. So every microbial genus will have many number of species, and each species will have number of strains. Among those strains, some can produce the disease, some can't produce the disease. So the strains which are able to produce the disease are known as virulent strains, and whereas the strains which are not able to produce the disease are known as avirulent strains. If you see the species, each species will have many number of strains, and among them some are virulent strains, some are avirulent strains. But to some extent, one species produces the disease which is also known as known as the pathogenicity. Say, for example, if you take the Staphylococcus aureus, the Staphylococcus aureus will have the number of strains. Usually, we say that Staphylococcus aureus is a pathogenic species. But again, the Staphylococcus aureus will have the number of strains. Some can produce the disease and some can't produce the disease. So the strains of Staphylococcus aureus which are able to produce the disease are known as virulent strains of the Staphylococcus aureus, which are not able to produce the disease are known as avirulent strains of Staphylococcus aureus. But basically, Staphylococcus aureus is a pathogenic species. So now if you see, the virulence means the ability of a strain to cause a disease. Pathogenicity is known as ability of a microbial species to cause the disease. And the virulence determinants, another thing is the infecting dose also virulence determinant. That means when the microorganisms are entering into the body to produce the disease, certain number of the bacteria are required, or certain dose of the toxin produced by those bacteria are required to produce the disease. So to produce a particular disease, the minimum number of bacteria required to produce the disease is known as minimum infecting dose. Say for example, in case of Salmonella and Vibrio cholera, nearly 10 to the power of 2 to 10 to the power of 3 bacteria, 3 to 4, 10 to the power of 3 to 10 to the power of 4 bacteria. That means 1000 to 10,000 bacteria are required to produce the disease. Whereas in case of Shigella, simple 10 to 100 bacilli also can produce the disease. So this is the difference. The minimum, this is the difference in producing disease by the microorganisms. Don't think that even though one or two microorganisms entered the body can produce the disease. To produce the disease by a pathogen, certain number, minimum number should enter into the body to produce the disease. Next one is MLD, that is minimum lethal dose. That is minimum quantity of toxin produced with the bacterium uh, should enter into the body to produce the clinical manifestations is known as minimum lethal dose. Say for example, if you take some X toxin, that X toxin can produce the clinical manifestations if it enter into the body 100 nanograms. If it enter into the 70 nanograms into the body, that toxin is not able to produce the disease. That is known as minimum lethal dose. Next one is bacteriophages. Bacteriophage means the viral particles which can infect the bacteria are known as bacteriophages. And these bacteriophages will direct the bacterium to produce certain lethal substances. Say for example, in case of diphtheria bacilli, the bacteriophages are responsible for the production of diphtheria toxin, which is responsible for their pathogenicity. If the bacteriophages are not present in those bacteria, the diphtheria are not able to produce the disease diphtheria by the Corynebacterium diphtheriae. And another important virulence determinant is route of infection. If you take the Vibrio cholera can produce the watery diarrhea, you all know that, otherwise known as the disease cholera. To produce this cholera, always the Vibrio cholera bacteria should enter into the body by means of ingestion only. If you give this bacteria through the IV route or by means of inhalation, the person will never suffer with the disease cholera. So the route is very precise or very important in case of Vibrio cholera if you take an example. But in case of streptococci, it can enter the body by any route and produce the disease. And so here if you see that streptococci should not be considered as more pathogenic than the Vibrio cholera. Here streptococci and Vibrio cholera both are pathogenic, but if you give the Vibrio cholera by means of inhalation, it won't produce. But if you give the streptococci by means of inhalation, it can produce the disease. So here their disease producing capacity of these two bacteria are directed or determined by the route of entry. That's also very important to produce the disease for any microbial agent. And how to alter the virulence of microbial strain. So this each and every microbial strain will have its own disease producing capacity, and this disease producing capacity either can be increased or can be decreased. So increasing the virulence of a microbial strain is known as exaltation, and decreasing the virulence or disease producing capacity of a strain is known as attenuation. and how to exaltate the virulence of a microbial strain. If you passage the microbial agent serially in a susceptible host, this which where I'm showing in the photograph, if you give the microorganism into the susceptible host, that is here the mice, and from there to another mice, the microbial agent will act like a more pathogenic than the first strain. Attenuation means reducing the virulence by growing the microorganisms under unfavorable conditions or in an artificial medium or by prolonged storage or in the presence of weak antiseptics. If you would like to reduce the disease producing capacity of any in some strains, you have to grow these organisms either one of these conditions. So they loses their pathogenic property but retains their antigenic property, that is known as attenuation. The best example for this is BCG, production of BCG vaccination. So if you grow these mycobacterium strains which are used for the BCG preparation of BCG vaccine under the unfavorable conditions for about 231 subcultures in a culture medium, but in unfavorable conditions. In unfavorable conditions, the microorganisms will grow but loses their pathogenic properties, and these bacteria are used as vaccine in the form of BCG. And the types of infectious diseases, and depending on the location, we can call them as either localized infections, which are localized infections may be superficial or deep seated. Generalized infections may be present in three forms. One is bacteremia, means if the bacteria are present in the blood, it's known as bacteremia. The bacteria which are present in the blood, if they are able to produce the toxins, now the bacteria plus toxins are present in the blood, which is known as septicemic condition. And if these bacteria and toxins, if they are able to produce the multiple abscesses in the internal organs of the body, then that is known as pyemia. The bacteremia is not that much dangerous, but septicemia is bit dangerous, which causes for the septicemic shock, and pyemia definitely is in a fatal condition in any individual. And types of infectious diseases, and depending on spread, uh, endemic disease means if any disease persistently present in low levels in a particular area, then that disease is known as endemic disease. For example, typhoid and malaria. If you see in the community, continuously you can see one or two cases in a particular area almost every day or maybe every week or something like that. And another form of the infectious disease is known as epidemic. If the infection which occurs higher rate in a particular period, then it's known as epidemic. Say for example, influenza and adenovirus, and these viral infections are seen in the community in large numbers and a given or particular period. Say for example, if you take the November, December, or January, you can see many number of common cold cases produced by either adenovirus or any other respiratory viral infections. That is known as epidemic. So infection which occurring in higher rate in a particular period is known as epidemic disease. And pandemic means infection that spreads rapidly to many parts of the world, either through water or through the air, and that's known as pandemic disease. You all know that the swine flu, bird flu, cholera, all these are pandemic diseases. And now, even COVID-19 also is another pandemic we are considering. So the pandemic disease, it starts in a particular in one place in the world, and total it encircles the entire world and produce few or many number of cases in every part of the world. That is known as pandemic disease. And so how to avoid infections? So either in the hospital or anywhere, limit visitors or no visitors, especially a person who got admitted, your kith and kin when got admission in the hospital, don't allow the visitors. Otherwise, the visitors may carry the microorganisms and they may transmit to the patient. And already is a patient who is suffering the disease and causes for the these organisms which are transmitted from the visitors can cause for other infection, otherwise called nosocomial or second infection in the individual. So they have to be careful about this one. And so don't disturb the normal flow of the body and save me. This is the future generation request to all of us. So if the even in the normal flow of the body, if you damage by means of indiscriminate usage of higher antibiotics, may responsible for the decreased immunity and which responsible for the disease manifestations even by the commensal organisms which are present in the large intestine. I think this is these are the some questionnaire for you people uh, to work out. So this I am giving the here the answers also. I am disclosing the answers. So with this, you just work out and have an analytical thinking about this particular topic. And this is about this particular topic. And thank you all.